And let's see if this works. Okay, that doesn't look bad. First of all, there is more information in the portfolios, and you can see under presentations that we have the presentations we started last class, the number 1, there's number 3 and 4, and I'm going to name them or number them accordingly so you don't have any problem finding them when you need to study. Please, I usually try to take notes of everything, but take notes of what is being covered in the quarters or in the month so you can study later. Now, in that regard, I also added a couple of pieces of information back there. That calendar, which contains information such as when the tests are going to be. So I'm thinking, or actually I set that down, that on February the 8th, we'll have the first one. Then March the 8th, the second, April 12th, the third, and May 10th, which is the last day of class, that will be the last test. The recovery, of course, it's assigned for the date as the final, and those I do not assign, so I don't know when they're going to be. It could be the 13th, which is the next Monday. It could be a week and a half later than that, but that is going to be announced around March or April, okay? I'll let you know if I know about that. I'm going to be absent from the 8th to the 10th on April, so that's going to be 2 classes that we won't have, and it's already in the portfolios, that information. You need to check it. Yeah, you can ask me, of course, but because I wrote that down, I'm going to easily forget. I'm going to check it anyway right here. Any questions? If you need something outside of the classroom, please email me. I sometimes sit for hours on end on my desk, but sometimes I'm nowhere near the department. But if you need me, just contact me and we can arrange for a schedule for you to visit. Let's see. Yeah, well, we already been through this. The tests are going to be on the computer, so the regulation about pencil and pen doesn't matter. And last class, yeah, we covered the generalities about databases and search engines. So a couple of classes I'm going to guide you through other parameters, but it's going to be both some theory, if you will, and we're going to try to practice. And I'm going to bring different examples, and I'm going to try to cover clinical biochemistry as well as biology. But again, if you have a suggestion, question, please let me know about it so I can improve the class as we move along. So first, database, UniProt or XPASI. It has those, not 2 names, but actually 2 different phases. I prefer XPASI, but that is just a matter of preference. Then we have these items that I have organized in order to try to help you recognize different parts of a database. And here I'm talking, of course, about not only the database, the data itself, but the way it's presented and accessed, okay? So first, accessibility, location of the information, that is, once you open the interface, how does it look? Accessibility, do you know this concept, mirrors?
Different servers?
That is right. The concept of mirrors in computers is that there's several computers with replicas, exact copies of the same information. For example, Google is not only one address in the internet, it's several addresses. I don't know even how many. The UDLAP is only one. Hotmail, any engine, any resource on the cloud that requires tons of access is going to have several addresses. It's going to have several mirrors. We're going to see some databases where that mirroring is pretty obvious. Some is really not that obvious, but it works in general. Yes?
Could you please repeat the definition of mirror?
Well, no, it's not a definition because this is not computational biology or computational systems. But the concept of mirror is that there's replicates of the information in different physical places as well as internet spaces. So for example, each one of these computers could be a clone of the other. It's a mirror copy. Well, not a mirror in the sense that it's the reverse, but it's the same information over and over. But here in this physical location, it's not that useful, except that you could access the same programs and the same system and whatnot. But across the globe, it means that if you are living, let's say, in Australia, you don't want to connect on a server in Hawaii. Oh, well, maybe actually it's close, but you wouldn't want to connect to Mexico, right? You would like to have your search go as close as possible. Maybe New Zealand, maybe the same Australia. And that is accomplished through mirrors.
And it can be on the internet too.
Yeah, it's actually on the internet. It's like an old definition of a cloud, right? Where is the cloud? It's not only one cloud. It's actually mirrors, and so on. And here I added this trace route. In your computer, if you access the command prompt, you can type something interesting. Let's say I haven't tried this in a while. Let's see if it works. Let's try utlap. Oh. Yeah, I don't know how to spell apparently. If you type trace RT in that command prompt, it's going to let you do what I'm going to do here. I have a different command. And the outcome of that command, as you can see, is an internet address. But because I'm connected to the network of that address, it's not going to give me other information than that I'm connected to that address. Let's try, as I said, Google. So you can see there's another internet address. With this trace route, actually it doesn't show it for Google, but it could tell you, I'm going to pick one of the many addresses. Why? Because there's mirrors for that address. There's copies of the same search engine. Oh, there we go. I'm not going to teach you how to read that, but there's information about how different is the internet to a telephone line, where you could call to one person. Here, as you can see, it has to go through several computers. And some of these computers are inside the university, others are outside. Okay, so that was just a little simple demonstration. It's not that important. A good database must have flexibility in the search terms and the logic operations. And it will force you to use not, and, or it could let you just do it through the GUI without having to think about those operands. An example is Google again. If you start typing words or adjectives or nouns, it's gonna assume that it's and, right? If you type shoes, sports, and bright colors, it's gonna assume it's and. And if it cannot find that logical operation, and it's gonna try and, or, and removing pieces of the in the search. So much so that it's going to tell you, did you mean that? Or this? Or these are the searches without this term. You don't have to know the operands, right? The search engine does it for you. Okay, but besides the search, the quality of a database is also important. What is the validation of the data? Who revises the data and makes it work? Here there should be, under etc., an annotation. Why is that data important? What is the correlation of that data with other subjects? Versioning and history. How long has this data been kept? When was the last time it was updated? And how old it was, or it is right now, without changes, for example. It's probably useful to have a way to compare its internal validation to biological relevance, right? This is kind of opposite to what happens on Wikipedia. On Wikipedia, you can type whatever you want. Maybe somebody will look at it and curate it, but nobody's going to go back and check the science behind what you wrote, right? But in a good database, we actually want that. And also to experimental methodology, right? You would like to know how that information came about. Is it somebody that sets that in a paper, or actually perform experiments published in that very same paper? So here, there's another last set of items or characteristics, and transparency. knowing what's in the database either when it's remotely or locally. It should contain citations, searchable categories, consistent results, right? Looking up the same term should always give you the same information unless the database is updated, unless something changes in the database, not in the algorithm underneath. And usually, because we're humans, not machines, we would like to have shortcuts to items, to categories, to things that we can organize our own mind with. So let's use this data. And if you please open another web page, another tab, and go to UniProt, get Sunheit, and look up for triose phosphate isomerase and Escherichia coli. I think I spelled them correctly, but feel free to correct me if I'm mistaken. So I'm going to open my own. There we go. Triose phosphate isomerase Escherichia coli. A good database is also going to be able to deal with misspellings, but try to spell as accurate as possible. Oh, so what did I do wrong? Yeah, big misspelling. There we go. Does your result are like mine?
Yes.
Just like Google, this database is telling us how many results it found. Although it's a little bit cryptically. If you look over here, let me find my pointer. If you look over there, it says 235 results. And it also says it right here. But it's broken down into two categories. We'll talk about those categories in a second. So let's go item by item. Is the information accessible? Well, so far, it's not that hard to read, but not that easy to read either. Even though we look up only two terms, the name of an enzyme and the name of an organism, our results include an entry number, an entry name, one of the search terms, gene names, an organism, another search term, and a number. It doesn't say what the number is, why do we have a gene name, why do we have an entry name, but we'll get to that. At least the search terms are obviously there. The name of the organism and the name of the enzyme. Okay? Sure? Everyone? Okay. Let's try to evaluate all of the items. Can we look using these operands? Just like the example for Google, the way we search was AND. Let's put in between E. coli and the name a huge not with capital letters. And click enter. Yeah. No, don't worry. That's why we're here, to learn, right? So the results are actually significantly different. It's only those results that look blue, according to the filter on the left. And there's only 10, as opposed to the 325 that we'd have before. So it's clear that without any term, you are searching for and if you want a not, you need to put it right there in between the terms you want to exclude. For example, what will be the result if we reverse the search term? Let's put Escherichia coli first, not triose phosphate isomerase. because these operands are logical, that the results should be dramatically different, right? So what do we find? That we have 31,000 results, probably mostly Escherichia coli, although because we have different strains, we might have more than one Escherichia coli listed. We have 1,200 results in the yellow and the rest in blue. So clearly these logical operands are actually doing something, and if we understand them, for example, this search was everything that's Escherichia coli except triose-phosphate isomerase. It's excluding only one result. In the opposite, it was everything that is named triose-phosphate isomerase, but it's not simultaneously E. coli. So at least that accessibility is there, right? We can add the and, not an or, but also we can do it through a GUI. And the order of whatever we're looking for is going to affect what is going to happen.
What is a GUI?
Graphical User Interface. Pretty much everything that is not just text. It could be a very primitive GUI, but... Oh, sorry. Here, one of these slides is not up to the date. Now, this is just the search result. I'm going back to the original one, the one that has an implicit and. I've checked that in my case it has a typo. There we go. And we are now confronted by this, the fact that we have two Escherichia coli results. If we know which strain we prefer, either the K12 or the DH108, we have to make that decision. And we can scroll down and see that, in fact, we have other Escherichia coli strains represented here, actually quite a few. And we have to decide which one we need to use. Okay? You already seen me scrolling, and some of this text in the table actually gets highlighted, right? So that means, as you know, you can click on it and get somewhere. So let's take exactly the first result. Oh, and by the way, take note of that number in the first column, the entry. I'm actually gonna write it down. I haven't memorized it. Let's see. Here. Okay, P0A858. I'm gonna click right on the entry number, and we're gonna be transported to this page. Now, the organization of this page is actually quite interesting. It starts always with UniProt, KB for knowledge base, the very same entry number, and the entry name. This is information that was in the columns, and it goes then into the name of the protein, the name of the gene, and the organism of origin. So even if you accidentally click here, you can still see all of the information that was on the table. It has a list on the left of pretty much all of the information that is contained. This is going to be included in accessibility and the flexibility, because we sometimes might be looking for a specific piece of information and we don't want to read through everything, right? We don't want to spend 30 minutes just trying to find where does it say when was it first purified, or if it's a protein, an enzyme, or whatnot. So what you can do is simply look up the data that you want. Function, name, subcellular location, pathology. You know what PTM stands for? Post-translational modifications. Those that have already taken cellular biology, everything that happens after the protein leaves the ribosome. Phosphorylation, glycosylation, and so on. Expression, just the production of RNA from DNA. Interaction, structure. Here are all the categories. Something that you probably won't notice until you find a very different protein is that the fact that all of these entries on the left contain a check mark and look blue means that that information is available. Let me go back, and I'm going to select from this 208 entries. I'm going to click on that, and then I'm going to click on one. It doesn't matter which. But look what happens here on these entries. There's at least 2 that are gray out. So for this entry, those three items are not available. There's no information that relates to this protein for this, what is it? PTM, pathology, as well as expression. There's no reports, there's no information. And you can quickly know that without having to scroll down and look for it. I'm going back to the one we have selected previously. And notice what you can do. So I'm here at the beginning at function. I'm going to go and click on sequence. It didn't send me to another web page. It only moved me ahead, as an index should do, to the place where that information is located. I want to go back to function. All I have to do is click on function. Right? So it's pretty accessible. It's pretty easy to move and to find the information you want. There's also this information that we kind of skipped, so I'm going back to the search. Of course, if I can. Here we go. So this view by is located on the side. It's a little bit lower down, but it separates the results table, a taxonomy, ontology, enzyme class, and other pieces of information. Let's say you don't want to see the entry, but want to see the relationship of this entry with other aspects of biology. And you can see how this is pretty much driven by biology, taxonomy. Ontology, you know what ontology is? No? Well, it's similar to taxonomy, but it has to be related, one to populations, another to a lower class of groups. Pathways, biochemistry too. You want to look up the pathway. You don't go into the enzyme entry. You look at the pathway. Where does the triose phosphate isomerase should be? Well, it should be glycolysis and glyconeogenesis. Go back. I keep going. This typo is gonna follow me forever. Now, what about the validation, the curation, and the annotations? Well, we have 2 levels. Out here, we have these 2. Reviewed or unreviewed. And if you read carefully, it says records with information extracted from literature, that is the available original source of information, and curator evaluated computational analysis. That is actually 2 things. A computer did an analysis, and then somebody checked the analysis. In my opinion, this is the highest validation just for the data itself. Then we have the blue one, which is unreviewed and stands for records that await manual annotation. So that is, these come from the literature and a computational analysis, and that's it. They still haven't been checked, validated by a human. In general, unless you want as much information as possible, you will go for the reviewed, because they are going to contain the most information and with the best quality. But there's some instances where you want unreviewed. And let's talk about examples. If you want to compare the human genome versus the orangutan genome, gorilla genome, you go to reviewed. But you want to compare strains of influenza virus, dengue virus, chikungunya, something like that, you're probably going to find them in unreviewed. Those genomes are sequenced frequently, and they are updated so often that they are not all curated. And if you want to try it, just type influenza, I don't know what, H1N1, and you will see that it's not going to be only one result, but it's going to be several, and it's very likely that it's going to be unreviewed. So that's one level. If you click again, or if you stay on the On the entry, you will notice that again we have the review, the standard versus unreviewed, but then we have these dots, annotation score, specifically the annotation, and here the score is the max, that is the 5 dots are full, and it specifies there's experimental evidence that that gene expresses as a protein. Other levels, let's see, can we see them here? Here are the levels. Protein uncertain, okay, it may be a protein. Protein predicted, it's similar to other known proteins. Protein inferred by homology. Oh, there's something very similar that is known to be a protein that looks like this. Experimental evidence at transcript level. The mRNA has been detected. And protein level. So for some instances, you may be satisfied with the transcript level or the homology level, and for some others you really want protein level, the maximum. But there's a way to find out that. You don't have to just cross your fingers and hope that it's a protein, right? There's evidence or not. Oh, sorry. What about the history? Well, look right here. This is the history. If you click on that, entry version. It's been revised 122 times. Doesn't sound like a lot, right? But imagine if you were revising the human genome, about 3 billion letters. If you revise that 120 times, I think by now every mistake will be ironed out, right? Or maybe not. And the version of the sequence, the original version of the sequence, dates back to 2005. And even more so, you can click around here and see previous versions, how the annotation of this sequence has changed. That, for historical reasons, might be useless. You don't care about that. But you can see how this computational ethos or work ethics pervades into databases, right? You don't want to have only the latest, but the previous and the previous and the previous, just in case. And if I click on that, yeah, it's pretty sterile, but you can actually select which one you want to compare to which one. Now, this will be a very complex way to find out what changed when. You could do it, machines will do it easily, but here you can do it manually. What about the internal validation and curation to biological relevance? In a way, this is a little bit related with these sections. What is the function? Are there pathologies associated? Where is the subcellular location? And I'm just going to scroll quickly through it. Here in the function, notice the items involved in glyconeogenesis, catalyze stereospecifically the conversion of dehydroxyacetone phosphate to the glyceraldehyde triphosphate. There's a UniProt annotation and a publication attached, right? So this is not the computer saying, oh, it looks like that enzyme. There's actually a publication that provides this information. And notice, I like this a lot. You don't have to scroll all the way down or till the end to find the publication. You click here, and here you go. That's the reference. In fact, you can probably click on the abstract, and it's going to take us to PubMed, and you can find the paper. Of course, sometimes, how old is this one? 1998. Maybe it's so old that we won't have access online, but doesn't hurt to try. Down here it has actual chemical representations of the function, the isomerization step. So if you were looking for something that you know the structure of, but know the name, this will help you a lot, right? You can see, oh yeah, this is a saccharide. This is what I'm looking for. Has information about the enzyme kinetics, the pathway, glycolysis, and whatnot. There's other information that we're going to talk about later, more specific, more detailed, but suffice to say that because of these correlations and the fact that we have citations makes this database very useful. You can know nothing about this protein, but you can read through it. What is important for me? The function? Okay, there's a couple of references there. Is it important the pathology? Well, I'll skip to the pathology. And what references are here? Oh, well, there's one. Nice, right? What about experimental methodology? Well, in some cases, there's going to have to be through the references. But there's another aspect here, or another aspect, which is the structure, for example. Structure determination, eminently experimental. Here, if you click on structure, you're going to find this interface that last time I checked, you can click and interact with. So you don't need to install any specific software. As long as you have a mouse, you can take a quick peek. It's not too powerful. You can, I think you can select some things up here, but it gives you a quick overview. And it actually redirects you to other databases where you can find more information. So at least there is that internal validation and relationship to other experimental methodologies and outside databases. Okay. Let's stop at this point so that we don't have too much information that we cannot make good use of it. And let me just emphasize two things. Well, actually, let me emphasize one more. You want a database to work for you. You don't want to have to spend hours understanding how it works. So the search engine, it's what's going to save you. If it allows for a specific logic operands, you can search for pretty much anything with as much specificity as you can describe in a sentence. Then you want to have your results in such a way that you can make sense of them. One thing I didn't show you is that these columns have these arrows. You know what will they do? They will sort them. So if you click on them, they are going to try to sort by order, for example, increasing order or decreasing order. So if you have a search that gives you so much information that you don't know where to begin with, you can click on these arrows and order the organisms alphabetically, either starting with C or starting with A. But that's not the best. Part of the flexibility, if you look at this, says columns. Try to click on that. You can customize not the search itself, but the results. Every single column that we saw before has a check mark. If you want different ones or more, you can click here. And you can add quickly as many items as are important for you. So this goes also hand in hand with the transparency and the flexibility. I like what I'm looking in the results, but I want other information. For example, caution, or I'm looking for proteins that do calcium binding, or I'm looking for proteins that are involved in RNA editing, right? So you don't have to even think so much about your search results or your search terms that you cannot actually customize this, so you can look for what you want. You can save them with cookies in your browser. So if you save a different search right now in those computers, supposedly next class you come by and search again, your preferences are going to be saved. I'm going to cancel this one, and I'm going to leave this unchanged. And another thing that I really actually would like to ask you, is any daltonic present in the class? No? Okay. Because I have no idea how a daltonic would look at these colors. Here the colors are simple, but they are a very good aid. Either you have colors on the side or on the center, and they are not so similar between themselves that they can confuse you, right? You want something very reviewed, yellow. You can see the different rows and columns by the colors. I actually haven't tried to use a filter for Daltonism to see if this is actually a good way to look to the results, because the other for sure way to look at these results for everyone will be to have them in black and white, and that will be awful, right? Can you picture this in black and white? I think I could show it to you in black and white. Would you like me to try? Let's see, accessibility. Grayscale. Okay, in gray actually looks hard to look at. And oh wow. Woo!
Eso es como negative.
Yeah, well, in bright colors, negative. So please, if you know somebody that is daltonic, ask them about these colors. I think it's a good scheme. It's not too bright, it's not too confusing, and it should allow you to navigate. Let's see. Yeah, and download for accessibilities. One thing that you can do is without actually clicking on the entry, you can check mark them and be able to download the information. So if you want to read what it is here, or if you just want the information itself, you can do both. In my opinion, this is one of the best databases out there because the learning curve is not that high. It's not that hard. As long as you know some biology, you can manage it. Okay, so let's stop here, and next class we'll pick up here.